are with a session from here. We are with a session from Ankita madam. It's our routine that every Saturdays and Sundays we conduct certain sessions so that we get aware of the research going in life sciences. We get aware of the coming trends in life sciences, and now we are with a series of the webinars where we are trying to focus on the institutions that are helping the research scholars in some or the other way. So today we are going to have some important information regarding an institute called as BioCard, and today's speaker is Miss Ankita Kumari. So I would like to first introduce our today's guest, Miss Ankita Kumari. First of all, it's my privilege to have Ankita madam for today's program. So it's my privilege to welcome you for today's session, ma'am, through MBSI and from my side. We welcome you, ma'am.
Thanks, Megha.
And let me introduce our today's speaker. Ankita ma'am holds a master's degree in biotechnology, which indicates a strong foundation in her field. Ma'am presently is serving as a managing director of Biocard India Private Limited, a company involved in biotechnology operations, and along with being the founder and director for also the Explorogen Discoveries Private Limited. Ma'am started her journey from DMCH Darbhanga, followed by CNCI Kolkata, and worked for Chromus Biotech Bangalore. With over 10 years of cumulative experience, her journey has evolved from hands on laboratory work to overseeing and managing a team of over 60 professionals spanning across the entire country. Along with her professional details, ma'am is also known to have more than 15 national and international publications, and we are very happy to have you for today's session, ma'am. So now I would request Ankita ma'am to take over the session, and later at the end, 15 minutes, we will have a Q&A session. So over to you, Ankita ma'am.
Thank you, Megha.
Yeah, ma'am.
Good afternoon, everyone. Good afternoon to all the participants. And on the behalf of our company, I would like to express gratitude, and we are thankful to Microbiology Society India. We would like to thanks to Dr. Desmukh sir and Megha and other faculty members who has given us the opportunity to present about BioCart and the application of genomics.
Ma'am, you are not audible.
Yeah, just a minute, Megha. I want to change the slide. There's a
Yeah,
technical
yeah,
issue.
yeah, sure, sure, sure.
Is the slide visible, Megha, right now?
Yes, ma'am, it's visible.
Yeah. So before giving a highlights of what all application of next generation sequencing is and genomics is, I would like to take some moment and introduce BioCard's journey. I'll just take few minutes of time and walk you through like how we started BioCard and what exactly we do. Yeah. So BioCard has been founded by Vikram Subramaniam in 2011. We started with Sanger sequencing and we established our Sanger sequencing machine in 2013. Since then, in late 2014, we established our lab with Illumina setup and we have developed a capacity wherein we have performed end-to-end services, as in starting from extraction of DNA, RNA, library preparation, and bioinformatics analysis. In late 2017, because of the high demand from the researchers, we came across a challenges that after the sample processing in the wet lab, like how to do this bioinformatics analysis, though there are a lot of open source of pipelines which is there in like various website it is present, However, we have developed in 2017 our own pipelines of bioinformatics, which has comparatively which takes lesser time and gives the same quality of data. In 2022, we have installed MGI machine because of the high demand in next generation sequencing and to meet the researchers' requirement, which gives equivalent data as Illumina. So, currently we have MGI as well as Illumina platform for next generation sequencing. So today we have team of something around 50 full-time employees and growing clientele India and across India, outside India. I would like to add that BioCart is successful. BioCart has successfully completed 13 years, and BioCart is second oldest genomics organization. And also, BioCart is one among the few who have their own Sanger sequencing machine, Illumina platform, and MGI sequencer without raising any funds and also without any investors' support. So here I would like to thank BioCart team who work day and night and considered BioCart as their own place and treat each and every project as a special and treat each and every project as their achievement. I also would like to thanks to all the researchers and the scientists who have actually supported us in this organic growth. This is our infrastructure in Bangalore. So, like we do have branch office in Lucknow as well as in Delhi. This is few machine which is which we have put it here in slide. This is Illumina machine, MGI, and the Sanger sequencing. Apart from that, we have QIAcube for RNA extraction, automatic RNA extraction. We do have entire setups for MolBio Lab and other genomics services. So this is the server which I was talking in the previous slide. We have developed in 2017. So when we were developing the pipelines, we have developed this server also in 2017. So this is customized server. So we have installed the servers in different part of India, as in Agriculture Institute, Junagadh, AMU, and some hospitals like Artemis hospitals, and in Kolkata, several places. So how this works? Once your sample has been processed in the lab, so like it's difficult for people to travel to come to our lab and do, let's say, do all the analysis. So what we do, these kind of servers we customize for as per their requirements. So we have provided here some configuration. So this server we make in a format that as per their desired requirement, we will be putting all the software and the tools, and we can put in your lab and you can do the analysis as per your desire. So there will be inbuilt tools and the software will be there, and we will be giving 3 to 1 weeks of training. So you can do metagenomics analysis in this server, whole genome, exome sequencing, or any RNA sequencing. So these are few recognition which we have got when pandemic has hit. We got recognized for the to take the responsibility for COVID genome sequencing. So this is one coverage which we got from IGIMS. So that time we were working with IGIMS and we worked for more than 1,000 sample for COVID genome sequencing. And apart from let's working with the Bihar government, we also work with Uttarakhand and Karnataka government closely with the hospitals to perform COVID genome sequencing. So this is some few other recognition which we got from Biospectrum, Industry Outlook, and Innovative Zone. So they say BioCart is a company of the year, and also they have in a survey they have found that BioCart is one of the best company for bioinformatics analysis. Along with the growth of BioCart, we have started other two company that is explosion discoveries and gene mine. So gene mine particularly for the clinical research work, clinical diagnostic work we have started, and explosion discoveries we have started wherein we manufacture all the different kinds of kit, let's say genomic DNA extraction kit, RNA extraction kit, NGS library preparation kit, and magnetic bead for DNA or RNA purification. So why we started explosion discovery is another story. So like a lot of researchers after basically after the pandemic, so when they were getting the kits from outside India, so it was like they has to wait for 3 to 4 weeks for the kit to get arrived and they need to start the process. So here in explosion we deliver the kits within 38 to 48 hours and with the equivalent quality. So this has reduced actually the turnaround time of the processing of samples. So these are few services which we actually do in BioCard: the DNA RNA extraction, microbial identification, Sanger sequencing, whole transcriptome, a small RNA sequencing are there, RNA sequencing we do, as well as chip sequencings are there, exome sequencing we do. Also some customized project if you have that we can like plan and discuss and execute. So before I actually introduce other various services, what all we operate in BioCard, I would like to start with the basics, evolution of sequencing, how it has happened. So everyone knows that first commercial sequencing technology was Sanger sequencing. Though there are a lot of disadvantages in Sanger sequencing, like you can sequence only the homogenized PCR product, and the sequence reads should be 800 to 1500 bases. However, still Sanger sequencing has been used for the validation of next generation sequencing results. And there are a lot of next generation sequencing platform. For short read, Illumina is there, Iron Torrent is there, MGI is there, and for long read, PacBio and Oxford Nanopore is there. So there are a lot of advantages and disadvantages of all these platforms. However, when you say disadvantage, like a lot of people say there are error in when you choose Nanopore or PacBio. But these errors are negligible because your sizes of products are longer than 8.5 to 10 kb. And usually we do all this in clinical samples. When you say short read sequencing, so there are Illumina and MGI, which deals with 150 base pair paired end, and Iron Torrent, which deals with 200 to 400 base pair paired end. We will see the application in later on the slides, like when to choose which platform while doing applications. So Sanger sequencing, what actually we need. So when you, let's say when you start any Sanger sequencing, we always ask you for unpurified PCR products. So wherein you do the genomic DNA extraction by yourself, no matter you use your phenol chloroform method of genomic DNA extraction or kit method of extraction, you amplify the product using the specific primers or targeted primers. You once you do the PCR amplification, we ask you to give 25 microliters of product, which we load on the gel electrophoresis unit to check the quality. If it's double band, we will be doing gel extraction. If it's a single band, we will be doing column purification or magnetic purification. So turnaround time for Sanger sequencing is 24 to 36 hours based on the load of the samples in the lab. And deliverables, I can say 800 bases of plus, which we deliver as a ABI, PDF and FASTA format. So here comes other it's a continuation of Sanger sequencing, microbial identification. Again, we do this microbial identification on Sanger sequencing platform. So here what happens? So we will be needing culture plate. or pure culture plate basically for bacteria identification or fungal identification or microbial identification. Even we can use for the plant sample or any tissue sample, you can say ant sample, all these kind of samples we can do the microbial identification using the specific primers. Let's say 16S we use for bacteria, ITS we use for fungus, SCO, LCO we use for parasites, ants, and 18S for the yeast. So, like we have a certain universal primer for all these identification. And when I talk about turnaround time, it takes 3 to 4 days time for giving us you the complete report. And as a deliverables, we give us a in this process, what actually happens is we do genomic DNA extraction, we keep PCR with a specific primer, and after that we do purification of that product, and we keep it in we do the Sanger sequencing of that product, and we make config of forward and reverse, wherein you get data something around 12 to 1500 bases, and we blast the result and get 10 nearest neighbor organism, and we share you the phylogenetic tree as well as we share you the ABI PDF FASTA of the raw file. So this is metagenomics, which comes under next generation sequencing, which I was talking in my previous slide. These sort read, which is Illumina, Iron Torrent, MGI, PacBio, and Oxford Nanopore. So these kind of platform we use here. So what happened in next generation sequencing? So there are a lot of services which falls in the next generation sequencing. That is metagenomics is the first one which I want to introduce. So any kind of environmental sample, as long as we will able to extract the DNA, let's say soil sample, water sample, swab sample, saliva sample, or any blood sample or plant sample. So what happened here is, after the process you will get to know the genetic information and the diversity of any environmental sample here. So a lot of a lot of bacteria is unculturable. However, in this process, you will able to find out those bacteria as well. So actually what happened in this process is, so platform which we use is Illumina MySeq. We generate data of something around 0.1 to 0.5 million leads. So if you are looking for bacterial diversity, we will be amplifying the genomic DNA with V3, V4. And if you are looking for the fungal part, we will be amplifying with ITS. So in this we will disadvantage, we will not able to target the viral or other part. Only we will able to tell you the 16S and the ITS part. And if you want to give the sample, you need to give us the raw sample, that is soil, water, or any environmental sample. Or if you want to extract the DNA, you need to give us 5 microgram of genomic DNA. And when it turnaround time is, so BioCart is known for lesser turnaround time for metagenomics all across India and outside India. That is 10 to 15 days. I think a lot of our researchers has joined this meeting who has sent metagenomic sample, and they must have observed that also, like 15 to 20 days we have delivered the entire report. So as a deliverables, what we have given is alpha diversity, beta diversity, which tells diversity in the sample, diversity among the sample. We also test the comparative analysis. We give the heat map and the other pathways. And if someone having the customized analysis which we need to do, they can always come up with a reference paper, and we are happy to serve equivalent kind of the research. So here comes whole metagenomics. So here in this whole metagenomics also, only the environmental sample will be needed. However, I told you in metagenomics, we will able to only give 16S or ITS, that is bacterial and the fungal diversity. In whole metagenomics, you will able to get algal diversity as well, bacterial, viral, fungal, and protozoa diversity. All these things you will able to find out. So nowadays, we have found that a lot of researchers are interested in knowing viral diversity. So for that, you can use whole metagenomics. So in this platform, which we use is Illumina HiSeq or NovaSeq platform, wherein as a standard, we will be generating data of 5 GB, which is 25 to 30 million beats. And as a analysis, we will be giving you the entire diversity along with the taxonomic profiling and the functional annotation. ARG study is one of the thing which is actually right now coming into the picture. So antibiotic resistant gene also we can tell you in this. And we have lot of publications which researchers has did with the biocard in antibiotic resistant gene using this whole metagenomics. So third comes here is whole genome sequencing. So in this, so what kind of sample is required for whole genome sequencing? So we always need pure strain for whole genome sequencing when you talk about bacteria and fungus. So for bacteria, 1 GB data is sufficient for publishing your paper, and for fungus, 3 GB data we usually give. And when you talk about plant or any plant, tomato plant or brinjal plant, so based on the size, we will be telling you how much data is required, 30 GB or 20 GB, whatever it is. And a lot of people are even interested in human genome sequencing. So since human genome sequencing sizes are big, so 100 GB data will be required for this. And here also same, we always prefer to get the culture plate as a confirmatory test. We will be always doing using the Sanger sequencing. The desired bacteria we will be telling you by showing you the phylogenetic tree. If the desired bacteria is coming via Sanger sequencing, then we will go ahead with the library preparation, and then we will be generating the data. As an output, we will be giving you the assembly, and this variant SNP we will be giving you, as well as the functional annotation and the alignment we will give it to you. In this also we can give you the antibiotic resistance gene. So apart from the DNA study, like in DNA, you can consider this chip sequencing also, that I have not included in this slide. So apart from that, there are next generation sequencing which falls with RNA as well, that is transcriptome sequencing, that is mRNA and miRNA. So we can do you can say, if you want me to tell you the samples, so cell culture we have handled, brain tissue, any kind of tissue basically, and plant sample, any kind of tissue and the blood samples. So basically what we do here is we extract the RNA. If someone is interested in mRNA, we will be extracting that. If someone is interested in miRNA, that we will prepare the library of miRNA, and we will generate data of again standard as a 5 GB or 10 GB, and turnaround time for this will be 3 to 4 weeks using the Illumina NovaSeq or HiSeq platform. Even if someone someone is in the requirement of MGF platform, we can do that also. So here what happens, what we give as an output is, if you want to do any gene expression study, if you want to know what gene is getting upregulated and downregulated, and the differential gene, that is what we say in transcriptome sequencing. Okay. So this is something which BioCart has launched as per their wish, you can say, after working for 13 years for the mankind and the researchers. So BISI is a program, BioCart India Scholarship Initiative, which we started in 2021. So every year, 5 students we are doing free of cost projects for 5 students. So here some terms and conditions are required, like we always verify the portfolio of the students from where they are coming, and what actually they are doing. So based on that, we verify the backgrounds, and we take 5 projects which we do free of cost under this scholarship program. We do have training and intensive program for UG, PG, and for PhD students. And we have launched from previous year. We since we were getting demands for this, so we have to start this, that you can get your sample, and we will do free real-time hands-on analysis with our experts. So I think anywhere else this kind of facility has been not provided. This also we are doing just because a lot of scientists has supported us, and this is just what we can actually give back to them as we have all the facility in the inside the biocard. They get their sample. Usually they can spend some time with our expertise, and they do the starting from extraction, library preparation, and analysis here sitting in the biocard with our expertise. So the fourth is after completion of any projects, we give a free session, as in what a report say, what a graph say, what a Excel say. So we give a 2 hour of session every Friday and Saturday after completion of the projects, wherein they can ask their doubt. Also, we will be explaining their reports one by one in a Zoom meeting, and if possible they can also come down and sit with our expertise and understand how exactly we have performed their research. Okay, so as I mentioned you previously, also we support till publication, and okay. So from early this year, January onwards, we launched an online-based platform wherein users can do metagenomics analysis free of cost by putting FastQ files in the tool. That is like open available, I think, right now. And next, after metagenomics, we are going to put this whole genome platform also open for all. So he is Vikram Subramaniam, who is CEO of Biocard, who have started Biocard company in 2011. So with this, I would like to uh, like uh, just ask if you guys have any question related to any projects which you are like having in your mind and you want to implement, or if you have any question which I have just now explained in the slides related to next generation sequencing. Ask.
Thank you, ma'am. It was really a very informative session. Now, the session is open for question and answers. You can unmute yourself and raise your question.
Yeah. Thank you very much.
Or if unable to talk, you can text your questions in the chat box so that I can raise the question.
Hello,
Yeah?
am I audible? Yeah,
Yes, 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 Chanda ma'am. How are you?
yeah. Fine. Megha, how are you?
Good, good, ma'am.
I had a question for Ankita ma'am.
Yeah, ma'am, sure.
I have some metagenomics data, and the company has we had sent it out um, for analysis, and the data which we have, we are not able to open the files or even access the files, and it is not in FASTA format. So we don't whether you would be able to help us with that. Do you have such a service?
Yeah, sure, we can help. Like you can always you must be having a raw file with you, right?
Yes, we have the
Like when you got your
yeah,
metagenomic sequencing done from some company, they must have given you raw file, right?
they have given, yeah, which we
Yeah.
are not able to access.
Okay, no problem. You can send us that raw file, and like
Okay,
we can do the analysis. Like whatever standard analysis I can do, I will show it to you. And apart from that, if you want me to add also something in that, I can do that also. So maybe like
okay,
mail
ma'am.
ID is written here, as well as phone number is also there with both Vikram sir's and mine. You can send us, and you
Okay,
can always give me the reference that through MSI, and we will be
okay.
happy to help.
Yes, ma'am. Yeah. Another question I had, ma'am, you said about the scholarship. So when is it open? Like, or which is it
So
announced on the your website?
so I think right now it's not open. For previous year, it's already done. So always during the
Okay.
month of when the actually session closed for a student, let's say March and April, when their results come out, that time we start that scholarship program. So during the
Okay.
month of April or June, we will again put in our LinkedIn website.
Announce it. Okay. Thank you so much. Yeah.
Thank you.
Anyone else wants to ask the question? I guess, just a minute, ma'am. Yeah, I guess there are no more questions because the PowerPoint presentation itself was very explanatory and it was really very informative. So, with your permission, ma'am, can we end the session?
Yeah, sure, Mega. We can end up the session. If they have at all any question in upcoming days, they can write us, and we will be happy to write them back as an answer.
Sure, ma'am, sure.
Possible.
So, last but not the least, I just a minute, ma'am. Someone, Jagdish sir. Yes, sir. Jagdish Kumar sir, you have any query? If you are unable to unmute, you can text in the chat box. Okay, I guess no. So, I would like to uh, say thank you to Ankita Kumari ma'am for spending her precious time for today's session. So we are very thankful and grateful to you and your time, ma'am. And the way you gave the information about BioCart and all the process is really needed for today's research scholars, where we are indeed in search of the institutes which provide us such kind of our resources so that we can do our research in a very informative way. So we are really thankful towards you, ma'am. Thanks a lot.
Thanks, Mega. See you soon.
Yeah, ma'am. Thank you. And ma'am, I'll be circulating your flyers in MBSI group and all the groups related to MBSI so that anyone in need of any query can approach you directly through your mail ID or through your contact number.
Sure,
Okay,
Mega.
ma'am.
I will send you the brochure as well. And
Sure.
yeah, you
Sure,
can
ma'am.
say my thanks to Dr. Deshmukh sir as well.
Sure, ma'am. And even thanks to all the participants who have joined today for the session in their busy schedule. So thank you to all of you. Shall we end the session, ma'am?
Yeah, sure. Thank you.
Yeah. Thank you. Thank you, ma'am.
Thank you, ma'am.